I would like to start this presentation and welcome you all to this webinar called How are the in vitro GARD assays used in real life? Cases and applications for testing of skin and respiratory sensitizers. This webinar focuses on applications from 6 real cases and examples and challenges with solutions using the GARD assay and what SenzaGen can do for you as a customer. To present this, we have Dr. Andy Folleryd, our scientific liaison and co-developer of the GARD platform. Andy has been part of the development of GARD during his PhD at Lund University in Sweden, and he's now working for SenzaGen for a long time, and hence he has a deep knowledge of the assay. So please feel welcome, everyone, and I give the word to you, Dr. Andy Folleryd.
Thank you, Hannah, very much for the introduction. And I would, of course, also like to take the opportunity to welcome you all to today's webinar. I'm really excited to see that so many have signed up for the webinar today. So as Hannah also briefly mentioned, the webinar that we will have here today will basically be a follow-up for an earlier webinar we had in April, where the focus was very, very technical, and it described more the development of the GARD platform. So if you were not able to attend this webinar, there will also be a recording of this webinar present on our homepage. So today's presentation will focus less on the technology and instead try to describe different customer studies of customers that has actually used the GARD platform in order to address certain challenges associated with the testing of their specific compounds. As Hannah also mentioned, I will try to lead you through 6 different cases that cover the classification of difficult to test substances, different type of UVCBs, complex mixtures, and medical devices. So before I go into the specific details, I would just like to give you a very brief introduction to SenzaGen. So we were founded in 2010 as a spin-out from Lund University, and today we comprise a highly multidisciplinary team of 20 employees, including 9 PhDs. Our business model is very simple. We will continue to develop new in vitro assays at our facility, and then we transfer the technologies to different CRO partners and industrial collaborators who perform the routine testing for their clients. So our lead product today is GARD. It stands for Genomic Allergen Rapid Detection, and it's a state-of-the-art testing platform for assessment of chemical sensitizers. So this platform has today generated 4 readily available assays. We do have the GARD skin and the GARD potency assay for assessment of skin sensitizing chemicals. We also have the GARD air assay for identification of respiratory sensitizing chemicals, and now also recently we have the GARD skin medical device for skin sensitization assessment of medical devices. So we are a very research-oriented company. We continue to develop new in vitro assays, but we of course also work to improve the assays that we have available. So one thing that we work really hard on is to increase the applicability domain of our assay. So today we offer the testing also of more complex samples, for example, different type of agrochemical formulations, medical device materials, UVCBs, and pre and pro-haptens. And some of these compounds will be covered in the different cases today. So since 2017, we are now also traded on the Nasdaq First North on the stock exchange in Stockholm. And we have established collaborations with different partners in the US, in the European Union, and also in Asia in order to distribute and license our technology around the world. So what I would really like to focus on and really like to emphasize in this presentation is that GARD is not only a single assay, but it's rather a very versatile testing platform on which we have based several different applications. So the assays that I will present today is the GARD skin assay for identification of skin sensitizing hazard. We do have the GARD skin for medical device testing, which is also based on the GARD skin assay, but it's capable of identifying skin sensitization hazard also from medical devices. We will also briefly touch upon the GARD potency assay, which is capable of subclassifying chemicals, skin sensitizing chemicals, according to the potency classes defined in the CLP regulations. And on top of that, we will also briefly mention the GARD air assay, which is capable of identifying respiratory sensitizing chemicals. So one thing that makes the GARD assay truly unique in comparison to other in vitro assay is that we use genomic biomarker signatures instead of only looking at single genes or single biomarkers for classifications. So as you can see in the slides, within the brackets is the number of parameters that we test in each of our assays. And as you can see, it's comprised of a set of highly carefully selected biomarkers and not only on single data points. And I would like to come back to later why this is important. So the different GARD assays can of course be used on their own, but they can also be used in a combination in order to provide a more thorough risk assessment of your specific substance. So this slide is basically to show how you can combine the different assay. So what you can actually test your substance at the same time. You can test it for skin sensitization, you can test it for respiratory sensitization, you can also say if the compound is both a skin and a respiratory sensitizers, and you can also draw the conclusion that the compound is a non-sensitizer. And of course, if you have then identified your compound as a skin sensitizer, for example, you can also add the GARD potency measurement, which is capable to subclassify your skin sensitizer as either a strong sensitizer or a weak sensitizer. So before I go into the different cases, I would like to give you a very brief introduction also to the technology for those of you that are not really familiar with how this actually works. But the GARD assay use a combination of human relevant cells and combine this with the measurement of genomics and the machine learning. So we believe one of the most important steps in the construction of a mechanistically relevant in vitro model is of course the selection of an appropriate biological system that is capable of actually reflecting the endpoints that you try to predict. So we know for sure that both skin and respiratory sensitization involves the activation of an adaptive immune system, and therefore we believe that dendritic cells is a very good target for assay development in terms of these two endpoints, because the dendritic cells are essentially the key player that bridge the innate and adaptive immune system. So as a biological system in the GARD assay, we use a human dendritic cell line that is called Senza cells. And this cell line is very much similar to the response generated in primary dendritic cells. So of course, once you then have defined your biological system, you need also to find a way to see how these cells actually react to external stimuli, such as, for example, when it's exposed to a sensitizer. So the conventional way that many in vitro models have done in order to monitor this, the cellular response, is to look only on one or two biomarkers, such as, for example, the upregulation of different type of phenotypic markers. So in the GARD assay, we use a completely different strategy, because instead of looking at in the regulation of only one or two biomarkers, we instead measure many different biomarkers in what we call a biomarker signature. So this is just an example to show the 200 genes in the GARD skin prediction signature that comprise the readout of this assay. So what you see in the heat map is basically the different regulation of the 200 genes in the prediction signature based on if cells are exposed to a skin sensitizer or a non-sensitizer. So basically these genes that we have in our biomarker signatures, they are of course not only selected at random. So if we start to look at the identities of the genes that we have in the GARD skin prediction signature, for example, we can see that they do not only explain one single mode of action, but they are rather involved in a number of different pathways that are known to be relevant for the development of skin sensitization. We have, for example, genes that are involved in the recognition of foreign substances. We have genes that are involved in the immunological self-defense mechanism and in the cellular stress response, and also that are responsible for chemotaxis and communication with other type of cells of the immune system. So in order to make this even a bit more, to explain this a bit more in details, it's also possible to map the 200 genes in the GARD skin prediction signature to the adverse outcome pathway for skin sensitization. So if you start to actually map some of the 200 genes to this AOP, you can see that we already measure, for example, CD86, which is the only biomarker measured in the HCLAT assay, for example. And we also measure those genes that are involved in the NRF2 pathway, which is basically the only endpoint that is measured in the keratinosense assay. So in fact, if we continue to actually map the genes, the 200 genes in the GARD prediction signature, we can see that they actually maps very well to the different key events in the adverse outcome pathway. And in this way, they actually form an integrated system to cover the major events that is associated with skin sensitization. So this is basically how we do in order to build more mechanistically relevant in vitro models. So based on this overall strategy, we have also defined a standard operating procedure on how we should test unknown testing substances. And this is, of course, a very broad simplification, but I'd like to use this in order to illustrate the concept on how we do the testing. So the first thing we do is basically to define the concentrations that we should use for the stimulation. This is called the GARD input concentration. We perform a dose response testing where we identify a concentration that induces a relative viability of our cells with 90%. So once we then have identified the GARD input concentration, we perform the cellular stimulations again at this GARD input concentration. We perform a quality control of the cells, and after that we simply isolate all of the RNA material. So after that, we measure the gene expression of the genes in the biomarker signatures, and dependent on what endpoint we look at, we measure the different biomarker signatures. So for example, for GARD skin that I used previously as the example, we measure the 200 genes and the regulation of these 200 genes, and we do this using the nanostring equipment. So the nanostring is a hybridization-based technology that is very similar to microarrays, but it enables us to measure only the 200 genes in a very robust and resource-effective way. So this raw gene expression data from the unknown testing compound is then uploaded into an online application. So in this online application, we have implemented a prediction model that is based on pattern recognition. And the specific pattern recognition algorithm that we use is called support vector machines. So the support vector machine basically compare the gene expression profile induced by the unknown testing compound to the gene expression profile of reference samples that we have in our database. So based on if the expression profile is most similar to a sensitizer or a non-sensitizer, the sample will be classified accordingly. So the interpretation of the results is very, very simple. So a positive value is classified as a sensitizer, a negative value is classified by the algorithm as a non-sensitizer. And I think that this is a very, very powerful technology to combine the human relevant cells with genomics and state-of-the-art machine learning. And just to briefly demonstrate some of the advantage with this data, I would like to show you some data from a recent validation study with the GARD skin assay. So I use GARD skin as the example because this is the assay that we have most data available on, and it's also the assay that is closest to have a formal regulatory acceptance as well. So we performed a validation study in accordance with OECD guidelines, and so this study basically involved a ring trial where we performed testing at at 3 different laboratories that were tested at our facility and also in collaboration with Burleson in North Carolina and Eurofins in Germany. So the result from this validation study demonstrate that this technology is very simple to transfer to other labs. The reproducibility both between labs and within labs is quite high, and this validation study also demonstrated an accuracy of 94% with a very good balance between the sensitivity and the specificity. So for this, the GARD skin assay, we have tested approximately 400 substances. Among those, we have yet managed to publish 207 compounds, and the accumulated accuracy of these 207 compounds is 89% for the GARD skin assay. With both the GARD skin and the GARD potency assay, we have submitted all this data to ECVAM, which is the European Center for Validation of Non-Animal Based Testing Strategies, and we are now currently awaiting the formal OECD approval. So in the meantime, we have a statement based on Europe with the European Chemical Agency that the results from these assays can already now be used for filling the REACH dossiers. So this is really good that we already have some kind of regulatory acceptance. So the next thing that I would like to briefly touch on is also, which I think is equally as important as the predictive performance of the assay, is of course to also evaluate the applicability domain of the assay. So this slide basically shows the distribution of the samples that we have tested among those 207 that we have published. So the bars correspond to the number of samples that have been tested within each chemical group, and the dots basically corresponds to the fluctuation in the accuracy across these different groups. So as we can see that regardless of which type of group of substances we are testing, the accuracy is quite close to the mean accuracy, which means that for this type of substances, the applicability domain is quite broad. So what I would like to also describe more in the different cases that I will soon start to go into is the testing of more complex samples such as UVCBs and natural extracts and mixtures, for example. So before I just start to briefly mention the cases, I would also like to mention that if you are interested in a more thorough discussion or introduction to the technology behind the GARD assay and how the assay was developed, but also to have some more data on the predictive performance of the other available assays, please have a look at our web page and under the webinar tabs, because there you can have access to the previous webinar that deals specifically with this type of information. So by that, I would like to start by describing a few of the cases. I would like to start with the first case that describes the use of the GARD skin assay for testing difficult to test substances. So the term difficult to test, of course, different across different industries, but in general, it is kind of used in order to describe substances that has traditionally been considered to be hard to test in vitro assays due to certain properties of the specific chemicals. So one thing that has been suggested to be quite tough to test in vitro system is pre and prohaptens, because they would require some kind of activation in order to create the allergen. So if we look at the gene expression of the Senza cells, we can see that we actually have quite high level of some of the known metabolizing enzymes. So in that sense, in theory at least, I don't think that it should be any problem to test these substances in our DC model. So the other thing that is often considered to be difficult to test is compounds that have a very high octanol water partition coefficient, or if they have other properties that will lead to a very low solubility in aqueous medium. So in general, I think that a potential benefit with the GARD assay is that we actually look at the transcriptome of the cells, and we can see these perturbations in the transcriptome at very low concentrations. So this means that it's sufficient to only have a very small amount of the test system, of the test item in solution in order to be able to provide a reliable result. So, but in order to thoroughly evaluate if we could actually use the GARD skin assay for this type of substances, we identified a subset of chemicals that were tested both in the GARD assay and also in the other OECD validated approaches for skin sensitization. And the selection criteria was based on if the substance was either a pre or pro-haptens or if it had a very high log p-value. And as you can see in this comparison, the majority of the compounds that were selected in this study was demonstrated to be quite difficult to test in the conventional in vitro assays. But one really important take-home message from this is that considering the pre and pro haptens, they were applicable to be tested both in the HCLAT and the GARD assay, which is the dendritic cell model. So I think this might be an indicator that these two cells actually have quite a good metabolic capacity. So if we look at the compounds with a very high log p-value and low water solubility, the majority of these substances could also be tested in the GARD assay. And I will come back to the description of how this actually worked later on in this presentation. But I think that the pre and the pro haptens and also the compounds with very low water solubility has been one of the main arguments for incorporating expensive and not very robust 3D cell models. And what I think and what I can actually show from this data is that the GARD assay and the HCLAT, I mean, the tools that are already available is quite applicable. Both the HCLAT and the GARD can, for example, be used for pre and pro haptens, and the GARD assay can be used for compounds with low water solubility. So that leads me basically into to the next case study where we use the GARD skin assay for testing of UVCBs. So the specific problem with UVCBs is often related to the very low solubility of this type of substances. They are often very, very hydrophobic. And that should in turn, of course, cause problem in assays that are based on cell media. So in this specific case that I would like to show, I would like to go into a bit more in detail on how we manage to test this type of hydrophobic substances that are not generally soluble in the conventional solvents, DMSO and water. So this few slides in this figure demonstrate basically the strategy we use when we test a very hydrophobic substance. So this is directly from a customer project. So we had a very hydrophobic substance, a UVCB. We first tried to get this compound into solution using either DMSO or water. And you can see from the top two figures to the left that this substance was not very soluble in the water or DMSO, and it was probably not even bioavailable to the cells, and we could not have any drop in the cytotoxicity neither. So if we look at the bottom two figures to the left, we can also see that this compound was incorrectly predicted as a negative in the GARD skin assay. So what we then did was that we went back to the customer. We asked them for a bit more information regarding the physiochemical properties, and we eventually decided to use a combination of less polar solvents in order to get this compound into solution. So we used a mixture of DMF and glycerol, and as you can see from the viability plot, we could actually have a bioavailability, we can observe a cytotoxicity, and this compound was then actually in the end also predicted correctly as a sensitizer. So we do today have quite a broad portfolio of different solvents that we have shown is compatible with the GARD skin test system. And regarding the last three ones, the hexane, the olive oil, and the sesame oil, I would like to show a bit more data on these specific extraction vehicles later on when we talk about the medical device testing. So based on the protocol with the testing of the different solvents, we were also sent an additional 7 substances from the sponsor of the study, and we could demonstrate by mixing around with the different solvents, we were able to correctly predict 6 of these 7 compounds in relation to the existing in vivo data. So I think that this study demonstrates that the GARD skin can be used for compounds with low water solubility, such as UVCBs, but it's also important to perform this type of testing on a case-to-case basis, because you really need to consider the physiochemical properties of the chemicals in order to select the appropriate solvent. So moving on to the next case, which describes the testing of natural mixtures. So this specific project is a collaboration between with the Federal University of Goiás in Brazil. And the aim of the study was basically to use a portfolio of different in vitro assays in order to predict the sensitizing properties of different henna-based hair coloring products. So henna-based products is often considered as a safer alternatives in comparison to the synthetic ones. But the majorities of these henna-based products also contains the known skin sensitizer PPD. And it's quite frequently that people actually develop symptoms by exposure to henna dyes. So this project was essentially divided into 2 parts. So first of all, it was performed a pre-study where a total of 10 hair dye ingredients was tested in the GARD assay and also in other in vitro assays. And in the main study then, the GARD skin assay was used to assess the sensitizing properties of 10 complex samples based on henna products, which was actually off-the-shelf and commercially available products. So, but just starting to describe the results from the testing of the different ingredients, we can see on the column to the right that the GARD skin assay was demonstrated to have a concordance to animal data of 73%, but it was also demonstrated to have a concordance to human data of 100%. And I think this is really good because it demonstrates how we would like to develop our assays. So I mean, both for developing our assays and for evaluating the performance of the assay, we use human reference data when available, because this is, I mean, what you try to predict is actually the situation in human and not the situation in an animal model. So I think it's more relevant to, of course, use the human data when it's available as the reference. So we also went on to test the 10 commercially available finished henna-based products, and all of these substances were classified as skin sensitizers in the GARD skin assay, which is in very good concordance with the predictions also from the other in vitro assays. And one thing that I think is quite interesting is that only 2 of the 10 compounds stated that they included PPD in their labels, but when our collaborators at the Goiás University actually perform HPLC on the samples, we could see that all of them actually contain PPD, but it was not labeled on the product. So, but it's really hard, of course, to tell if the sensitizing properties that were picked up in the in vitro assays if that is only based on the presence of PPD in this mixture, because it's a crude mixtures, of course, and it could also be other type of effects. But I think this case demonstrates that the GARD assay is well applicable to hair dye ingredients with a high concordance to human data, and that the GARD skin assay also should show very good correlation to other in vitro assays for testing of this commercially available mixtures. So the next project that I would like to show you now is also related to the testing of mixtures. In this case, it was a leading supplier of raw materials that is used in cosmetic ingredients or different type of personal care applications. So the problem this customer had with one of the oil-based mixture products was that it had a very unpleasant and very characteristic smell. So this was of course not very well received by the purchaser of this product, because it was used as a raw material that was sold both to the cosmetic and the personal care companies. So the thing is that this mixture contains specific chemicals. We can call it for chemical X as a contaminant, which was also the suspect of this nasty smell. So the company then applied a distillation process in order to get rid of this chemical X, and they tested all of these different steps in the GARD assay. So what we basically did was we tested pure chemical X, and we also tested, which is the test item 1 in this figure, and we also tested the oil mixture before the distillation process was initiated, which is the test item 2. And this was predicted, both this test item 1 and test item 2 was classified as non-sensitizer by the assay. And this also seemed quite logic, because these has been products that's been used been commercially available, and still nobody had complained about adverse effects. But when we tested the distilled product, the distilled oil, so the distilled oil did not give any nasty smell, but it was classified in our assay as a sensitizer, and that was also later confirmed by following up studies by this company. And what actually happened was that this company decided not to use this distillation process because it changed some of the chemical properties of the mixture, which resulted in sensitizing properties. So I think this is quite a good example on how you can use the GARD assay for quality controls of different type of production batches, and it also demonstrates that the assay is applicable for this type of mixtures. So the next project that I would like to also briefly talk about is the GARD skin assay used for testing of medical devices. So this is a very new assays that we now have available in our portfolio, and the testing is performed according to the ISO 10993 standard for evaluation of biological materials. So to comply with these standards, we have implemented an extraction procedure using both polar and non-polar extraction vehicles, and the intention is then of course to classify the leachables from the medical device in both of these two vehicles using the conventional GARD skin assay. So this is so all these procedures is stated exactly in the ISO standard on how you should be able to do this type of testing. So the first thing we did was basically to test that the GARD assay was compatible with both a polar and a non-polar extraction vehicle. So the figures to the left shows the testing of the polar extraction vehicle at different type of concentrations, and as you can see, you can use a concentration in well concentration corresponding to at least 50% of this extraction vehicle and still not have a false positive classification. And regarding the non-polar extraction vehicle, we tested up to 10% in well concentration, and it is possible to use also this solvent at a concentration of at least 10% without risking false positive classifications. And then the ability to detect the leachables from the medical device in both saline and different type of oil. In this sense, we use both olive oil and sesame oil, was evaluated using different type of silicone materials. We use a silicone tube and a TPE tube that was spiked with different type of sensitizers with different type of potencies. So as you can see that the pure material itself without any spike was classified correctly as non-sensitizers, and all the spikes in were also correctly classified as sensitizers in both the saline and the different oil mixtures, which demonstrate that this works really good. So in addition, we also tested 3 medical grade medical grade products, which is a silicone tube, a TPE tube, and a PVC tube. And these were also correctly classified as non-sensitizer in all the extraction vehicles that we used. So by that, I would also like to show you the last case study in today's webinar, which basically described how we use a combination of the GARD skin assay, the GARD potency assay, and the GARD air assay to predict the skin and respiratory sensitization potential of e-liquids. And as far as I know, this is actually the first published study. This is a collaboration that was performed together with Imperial Tobacco, and it has been published now as well. And as far as I know, it's the first study that demonstrates the safety profile of e-liquids using for the endpoint of skin and respiratory sensitization using only in vitro assays. So first of all, I think it's important to acknowledge that the composition of the e-liquids is a complex mixture, so it's quite difficult to actually test in vitro systems. So the e-liquids often comprise of a base liquid, which is generally the aerosol former, and it's also comprised of some kind of flavoring, and it's also often include nicotine, which is known to cause a problem in a lot of different cell systems because it's very cytotoxic to the cells. So from a toxicological point of view, the main issue with this type of e-liquids is basically the different type of flavorings, because they have in some cases shown to have allergenic effects. So from a safety point of view, it's really important to test these mixtures both for skin and respiratory sensitizing potential, because in that sense you can start to change one of the flavoring for a less dangerous alternative. So what we basically did first was to test the e-liquids in our GARD air assay for assessment of respiratory sensitizers. And in order to first demonstrate the performance of this assay, the GARD air assay for identification of respiratory sensitizers, in the figure to the left, we basically tested a proficiency set of chemicals with known respiratory sensitizing properties. And as you can see, the overall accuracy of this classification was 89%. So this kind of demonstrates that this assay could actually be used for testing this endpoint. So after that, we proceeded with the testing of the 5 e-liquids that had various composition. For the 3 samples to the left in the B plot, they contain only the base liquid formulation with different amounts of nicotine, which is stated within the brackets. And the 2 compounds to the right were the commercially available e-liquids that contained also the specific flavoring ingredients. So all of these compounds, the e-liquids, were classified as non-respiratory sensitizers in our assay, which I think is quite in line with the expectation from the customers, because these products has all been out on the market and people has been exposed to them without any adverse effect in terms of respiratory sensitization has been reported. So we of course went on to test the same 5 e-liquids also in the GARD skin assay. And there we can see that all of the base liquid formulations with different type of nicotine was correctly predicted as non-sensitizers. And we can also see that the 2 commercially available e-liquids containing the different flavors were classified as skin sensitizers. So we also continue to do the testing in the GARD potency prediction model with these 2 compounds that were classified as skin sensitizer, and they were all then classified as weak sensitizers, which in this plot is class 1B or category 1B. So one thing that I think is really important from the slide, at least for the GARD skin predictions, is that even though the base liquid formulation contained different type of nicotine, different composition of nicotine, and we tested this up to 4.5%, it did not induce a false positive classification in the GARD assay, which I think is very good. So I think this slide basically demonstrate that the GARD assay and how it could be used, and how the different assays of the GARD platform can be used together in order to inform both on skin sensitizing hazard and potency, but also on respiratory sensitization. So for this specific study, the safety assessment was performed on an e-liquid, but I think that the same strategy can also be applied for other type of similar mixtures. So that was all of the cases that I would like to show, and by that I would just like to briefly also give you the information on how you should do in order to be able to test your substances. The first thing is basically just contact us, and we will be really happy to help you to design a testing strategy that is suitable for your specific substances. You of course need to select the different assays that you would like to test, if you only would like to use one, or if you would like to use all the different endpoints. And we would be, of course, happy if you were able to also provide us with some information about the physiochemical properties, so we can select an appropriate solvent to use. If you don't have this available, we can also test empirically which solvents that is suitable. The turnaround time is between 4 and 6 weeks, so we have a very quick turnaround time. And we only require a very small amount of samples. We typically ask for 0.5 grams of solids and 1 milliliter of liquids. And this can of course also be adapted to much lower volumes if needed. And after that, you of course also need to select your preferred site of testing where you would like to have your testing done. And today we have the collaboration with 3 different licensed labs. We can perform all of the testing for you. We have a collaboration with 2 labs in the US, which is Burleson Research Technologies, that was also involved in our validation study. And they are based in the Research Triangle in North Carolina. And we also collaborate there with MB Research Laboratories. And in Europe, we also collaborate with Eurofins in Munich. And on top of the licensed labs that can actually perform the testing directly, we do also have a global net of different distributors that can help you to organize the testing at our different facilities. So by that, I would just like to finish off this presentation today, and I hope that I've been able to show you some new applications on how you can use this type of in vitro assays, and specifically how you can use the GARD assay for different type of difficult to test substances. And by that, I would be very happy to take any questions, and thank you very much again for your attention.